So this is the actual nephron. This is, well, this isn't an actual. This is a diagram of a nephron. It's a simplified drawing of the nephron, and what we've done is we've pulled the nephron so that it's very, very stretched out. And in doing so, you can see the renal tubule really well. So there's the glomerular capsule, Bowman's capsule, the renal corpuscle, whatever you want to call it. There's the glomerulus. This is where filtration occurs. All the filtrate moves into the renal tubule and becomes tubular fluid. So the renal tubule consists of the proximal convoluted tubule right here. The proximal convoluted tubule is not only the first part of the renal tubule, but it's also the area where the majority of reabsorption occurs. And the reason for this is because there's microvilli in the proximal convoluted tubule, and microvilli increase surface area for absorption. Fluid moves from the proximal convoluted tubule into this renal loop, or this loop of Henle. Okay, so here this is called the loop of Henle or the nephron loop, and it has a descending limb and an ascending limb. Okay. At the end is this tiny hairpin loop that moves the fluid from the descending limb of the loop to the ascending limb. Within the loops, there are regions where there's thin segments and there are regions where there's thick segments. So thin segments are going to be composed of simple squamous epithelium, and they'll be very good for diffusion of materials. Whereas thick segments are going to be composed of simple cuboidal epithelium. They're also going to allow for certain things to pass through, but they'll keep other things from passing through. And so this is where a lot of reabsorption of waters and salts occurs. And then we move to the distal convoluted tubule. The distal convoluted tubule starts at the end of the ascending limb of the loop of Henle, and it leads to the collecting duct. So this area also has minimal reabsorption and secretion because it doesn't have the microvilli that is found in the proximal convoluted tubule. But the distal convoluted tubule does lead to the collecting duct. So the collecting duct is where urine collects. So as the filtrate moves to the renal tubule, it becomes tubular fluid. But as it leaves the renal tubule and moves into the collecting duct, we have a large amount of water that is going to be reabsorbed, but the remaining material becomes the urine. And depending on how hydrated we are, we will produce very dilute or very concentrated urine. So the collecting duct leads into a region called the papillary duct, and the papillary duct then leads to the minor and the major calyx. The minor calyx leads to the major calyx, which leads to the renal pelvis, which leads to the ureter, urinary bladder, and urethra. So this is basic flow of fluid in the production of urine. Okay? So this is what a true nephron looks like. Much more complex looking. You have an afferent and an efferent arteriole going into the renal corpuscle, forming the glomerulus, or the glomerular capillaries. Here's the proximal convoluted tubule right here. Here's the distal convoluted tubule right here. And there's your loop of Henle. There's your collecting duct. So within the renal tubule, we have specialized cells called principal cells. Principal cells, you can see them in the collecting duct, are going to be sensitive to aldosterone and antidiuretic hormone. Aldosterone increases salt uptake. So sodium, I should say, uptake. Sodium moving back into the blood is going to influence water movement into the blood. So if we have a lot of sodium moving into the blood, we're going to increase the amount of water we reabsorb as well, which decreases the amount of fluid that is lost as urine. And then we have antidiuretic hormone, which increases the number of aquaporins that we have. Aquaporins are going to allow more fluid, more water, to be reabsorbed. So in both cases, we're going to decrease the amount of urine we form, and we're going to increase the amount of fluid in our blood. That protects us from dehydration. We also have intercalated cells. So you can see they're fewer and far between within that collecting duct, but they're going to be important in maintaining blood pH. So they regulate blood pH. If we have a very acidic fluid flowing through, they're going to respond by increasing the amount of hydrogen ions we release and decreasing the amount of bicarbonate ions we release in urine. We're going to reabsorb bicarbonate ions, and that's going to lower that's going to increase the blood pH back to where it should be. That's one of the ways that we maintain blood pH. Oh, wait.
All three of the harmful foods I'm going to show you right now are labeled.
I don't know what that is. That's not something I meant to do. I'm not sure what that is. I have a video on here, and I'm not going to play it because I don't know what it is. Maybe on the next video I'll play it, okay? There are two types of nephrons that we have. We have cortical nephrons, which make up the majority of our nephrons. About 85% of the nephrons that we have are cortical nephrons, and they're going to be high up in the renal cortex with a very shallow loop of Henle dipping into the medulla. And then the remaining 15% are going to be the juxtamedullary nephrons. Juxtamedullary nephrons are found by the cortico-juxtamedullary region or corticomedullary junction right at the edge. And so they dip real far down into the medulla. And the juxtamedullary nephrons are important in maintaining a salt gradient so that we're constantly increasing reabsorption of fluids so we don't lose fluids. Again, remember I told you that we filter about 180 liters of fluid, but we only have about 6 liters at most in our body. So we need to be able to reabsorb almost all of that fluid so that we don't lose fluid and, dehydrate, okay? Here's your cortical nephron with a very small loop of Henle. Here's your juxtamedullary nephron with a very long loop of Henle. So here's a video on the urinary system, specifically your nephrons.
The renal system consists of the kidneys, the connecting arteries and veins, and the urinary tract. The kidneys are paired, bean-shaped organs lying outside the peritoneal cavity in the back of the upper abdomen, one on each side of the vertebral column. The kidneys function as the body's main excretory organs, eliminating the body's metabolic waste products by filtering the blood. Substances that are unneeded or are present in excess are filtered out of the blood and form into urine, moving via the ureter into the bladder before being expelled through the urethra. The kidneys also selectively reabsorb those substances that are needed to maintain the normal composition of the blood. By adjusting blood composition, the kidneys are able to maintain blood volume and pressure, ensure the proper balance of sodium, chloride, potassium, calcium, hydrogen, phosphate, and pH, and eliminate products of metabolism such as urea, uric acid, and creatinine. The medial border of the kidney is indented by a deep fissure called the hilus, where the blood vessels, nerves, and ureters connect to the kidney. The kidney is composed of up to 18 lobes. Each lobe is composed of nephrons. Nephrons are the functional units of the kidney, and each kidney contains more than 1 million nephrons. Each nephron consists of a glomerulus and a system of tubules. The glomerulus is a unique, high-pressure mass of capillaries that filters the blood. The glomerulus is encased in a thin, double-walled capsule called Bowman's capsule. The space inside the capsule and surrounding the glomerulus is called Bowman's space. Plasma-like fluid is filtered from the capillary blood into Bowman's space through the glomerular filtration membrane. The glomerular filtration membrane consists of 3 layers of capillary wall: the endothelium, the basement membrane, and the epithelium. This membrane allows some particles from the blood to pass through, but not all. The fluid that is filtered from the capillary blood into the Bowman's space is called filtrate and forms the primary urine. The filtrate then diffuses across Bowman's space and into the tubule system of the nephron. In the tubules, some substances are added to the filtrate as part of the urine formation, and some substances are reabsorbed out of the filtrate and back into the blood. The nephron tubule is divided into 4 segments. The filtrate passes through each of these segments before reaching the ureter, a highly coiled segment called the proximal convoluted tubule, which drains Bowman's capsule and where almost complete absorption of nutritionally important substances takes place. A thin looped structure called the loop of Henle, which reabsorbs water and ions from the urine and plays a role in controlling the concentration of urine. A distal coiled portion called the distal convoluted tubule, which regulates potassium, sodium, and pH, and where further dilution of the urine takes place. And the collecting tubule, which joins with several tubules to collect the filtrate and where final sodium regulation takes place. Each kidney is supplied with blood by a single renal artery that arises on its respective side of the aorta, before dividing into 5 segmental arteries that enter the hilus. Within the kidney, each segmental artery branches into several lobular arteries. The lobular arteries further subdivide to form interlobular arteries, which branch off into afferent arterioles. Blood flows into the glomeruli through the afferent arterioles. Blood flows out of the glomerulus through the efferent arteriole. The afferent and efferent arterioles regulate glomerular capillary pressure by selectively dilating or constricting. The kidney's venous blood, now filtered, flows from the glomerulus via the efferent arterioles into the peritubular capillary network, a low-pressure, reabsorptive system surrounding all portions of the tubules. This arrangement permits rapid movement of solutes and water between the fluid in the tubular lumen and the blood in the capillaries. The peritubular capillaries rejoin to form the venous channels by which blood leaves the kidneys and empties into the inferior vena cava. Urine formation involves the filtration of the blood by the glomerulus to form an ultrafiltrate of urine, the tubular reabsorption of electrolytes and nutrients needed to maintain the constancy of the internal environment, and the secretion of waste materials. Filtration occurs as blood flows into the glomerulus from its afferent arteriole, and plasma moves through the glomerular capillaries into Bowman's space. From Bowman's space, the glomerular filtrate moves into the tubular segments of the nephron. Here, through tubular reabsorption, electrolytes and nutrients move from the filtrate back into the bloodstream. Here also, through tubular secretion, substances move from the peritubular capillaries into the urine filtrate. The filtrate concentrates in the collecting tubules, then finds its way to the renal pelvis, where it is directed to the ureter, the bladder, and the urethra for elimination. The kidneys perform an excretory function by filtering the blood and then selectively reabsorbing those materials that are needed to maintain a stable internal environment. The nephron is the functional unit of the kidney and is composed of a glomerulus, which filters the blood, and a tubular component, where necessary substances are reabsorbed into the bloodstream and unneeded materials are secreted into the tubular filtrate for elimination in urine.
All right. I'm going to shut this off because now I'm going to, bugger, did the same thing. I always do this. I'm going to stop the video now, and we'll start talking about the juxtaglomerular apparatus in our next video, okay?